Okay, I'm going to go ahead and get started. I'm going to pick up where we got cut off last Thursday. I looked over a few of the URLs that got submitted for the homework. Everybody seems, all the ones that I've checked, looks like they finished it okay. At least the alignments look like what I expect them to look like. So it seems like people were able to get through doing the alignments with muscle on bridges too. The steps where we got cut off are doing some visualizations. I'm actually going to sort of skip past doing that on the muscle alignments because we'll just do it on the MAFFT alignment. So step 10 was to download your alignments and then ask ChatGPT to show you how to do a traditional dot plot. Now, I know that ChatGPT didn't tell everybody how to do the same dot plot. So a few people went ahead and tried to do that anyway. And they didn't get exactly the same kind of plots that I was looking for or that I expected. They're still valid ways to look at a pairwise alignment, but it's interesting and I'm not sure exactly why. But there are lots of flavors of dot plot. So anyway, I'm going to show you one. And then the second one was to do a circular gene tree of those alignments, and I'll show you an example of that as well. And so we're skipping to this last step, which was to have ChatGPT show you how to do this in Colab using the other software package that I mentioned in the overview lecture called MAFFT. So this is the fastest. This is the one that most people use when they've really got lots of sequences to align, at least the one that I see in the literature the most. And because we're a little bit behind where I want to be, you get the pre-baked version rather than going through this step by step. So if you go over to Canvas, today's lecture has this link to a Colab notebook. So that should take you to this page. And if you try to run all from here, it won't work because it needs data. Okay, so mine's connected to a runtime, doesn't have any data in there, but first cell is, the first few cells are just installing the software we're going to need. So it's going to go out and collect the MAFFT software and install it. Shouldn't take too long. I'm going to hide that output. Next step is to install something called Trimall. It's one of many automated trimmers. So we'll talk about why that's useful when we get to that step. But this will go to the GitHub repo. So wget is a web get. And so it's going to go and download the zip file that we want and then unzip it. It's going to move the Trimall executable file, the actual code to run Trimall, into our path. That's what this move step is. And hopefully these commands sort of ring a bell from way back when we were doing Unix. And then RM steps to just clean up extra files that we don't need. So you may see stuff pop up over there in the left column as things happen. So it downloaded it and unpacked it. And then, they disappeared because after it unpacked it and moved it, it went back and erased it, the stuff that we don't need. And so if you were to navigate into user local bin, you would see that there's a new file there called trim all. And all that, those last few steps of moving it into the path and then cleaning it up, not necessary to run it. They just make it more convenient to run. So now we can just type trim all, and since it's in our path, it will know that's a command. Whereas if we don't do that, we've got to specify the whole path to the program before we do any options for that program. So it just makes the command line look a little cleaner, less typing in the long run. And then the last thing here for installs is to install BioPython. We're going to use some functions from BioPython for our plotting. So if we didn't get any errors, hopefully everything worked, but I always like to include, especially when I'm trying new software, version check. And so if you run the version check, that tells you not only important information about what version you have of each one, but it tells you that they all installed okay. Otherwise, you would get an error that BioPython wasn't found or trimall wasn't found, something like that. Okay, so we've got all of our software installed. Now we're just going to run MAFFT. But, bolded bullet here, it assumes that you've uploaded the P450 data. So you'll have to go find that on your computer, or you can re-download that from Module 5 in Canvas. I've got it saved locally, so I'm just going to grab my P450. Whoops, don't open it. I'm going to grab this and drop it into the files over here. So now I've got it in my runtime. I'll give you all a second to get that. I've also copied a link to the MAFFT manual there. MAFFT does have a nice web server. So if you just Google MAFFT, you can download this to your local machine, whether you're running Mac or Windows or whatever. I might have talked about this when I was talking about software. But in any case, there's an online server. And the reason I'm pointing this out is when you go to the manual, you'll see that it walks through all the options that you can put on the command line. And for me, a lot of times, I don't know, it makes more, I'm a graphical visual person, and it makes more sense sometimes when I'm looking at it in the GUI relative to just thinking about it on the command line. So basically all of these little tick boxes that you can check or uncheck are different options that you could put on the command line. And so by clicking this box, it's literally, are you going to put that option on the command line? So on the back end behind this GUI, it's generating the command line for you. Okay. Yeah.
Where did you download the P450 again?
Where did I?
Yes.
I just dragged it into this window. You can also click the upload button, so this little button, and then it'll give you a dialog to pick it from your machine wherever you have it saved. So once you've got that, you can click the run button. And so this command line just says run MAFFT, And auto means pick the algorithm based on the data. And so MAFFT, another nice thing about the GUI is that, right, these are all the modes that you can force MAFFT to run in if you want to. Otherwise, it will pick the one that is optimal for the size of your data. So if you've got lots and lots of sequences or a modest number that are really long, you might need to pick a faster algorithm that is not guaranteed necessarily to give you the optimal alignment. So all of that goes back to that whole lecture I talked about how you do alignment and how multiple alignment works. MAFFT uses, depending on which mode, can use both progressive and iterative approaches and combine them to try to get the optimal alignment. By default, it's using Blossom 62 as a scoring matrix. It also running in auto, it auto detected that it's aligning protein sequence, not not nucleotide sequence. And so it used the appropriate scoring matrix. You can see all of the options, the weights that it gave it, gap penalties and all that stuff we talked about for things that are important to alignment scoring. Okay, and then down here, the strategy, you used the standard, right? This is the FFT, fast Fourier transform, is the standard model. We've got a medium amount of data, actually kind of small. So it used what ought to be the optimal method, except, well, I should say optimal. It's a balance of speed and accuracy. You'll see in a minute that it'll use a slower method after we trim. Okay, and so you should then see that align.fastday popped up. If you wanted to, you could download that and look at it in the NCBI viewer, just like we did for the muscle. And in principle, and in fact, if you actually do it, I've done it, you get something that looks very much like what muscle gave you. Okay, so not too much different. So that's kind of where we ended with muscle. We've got our multiple sequence alignment for P450, and we use the sequence viewer to try to see, what does that alignment look like. Another way to look at the alignments is to do a dot plot, and this can tell you some interesting things that aren't as apparent just looking at the sequence viewer. So if you run this cell, it's going to generate these dot plots. And it should download one to your computer as well. You can comment out that last line if you don't want it to automatically download. The other thing that this code generates is a directory that has all of the individual pairwise. Well, we only ask it to do the top 16 most similar. That 16 is completely arbitrary. I just picked a 4x4 grid. You could do all against all. That would be a lot, we've got 120 something sequences, so that'd be a lot of dot plots. I think it's sort of informative to just do the best ones, because if the best ones are bad, the rest are going to be worse. Okay, so what you're looking at here, this uses a Kamer approach where it chops up the sequences into I think 9 mers in this case. You can adjust that. Yeah, so here K is 9. And it slides the window along and says, on sequence 1 and sequence 2, is the K-mer the same? It scores it, does Hamming distance, something analogous to Hamming distance. And if they're a good enough match in that K-mer, then plot a dot, okay? And so it will slide sequence 1 all the way along and sequence 2 all the way along, and everywhere that you get a K-mer match below that threshold, below that threshold, it will put a dot, okay? And so if the two sequences are roughly the same length and everything sort of matches window by window, you should see a diagonal of dots, right? Because as you go from 0 to around 500 in this case for these two sequences, actually let me click on that just so it's blown up a little bit. Right, what that's showing you is that as you slid windows along, you got best hits or the K-mer matches, not best hits, skipping to blast already, where these two sequences align really well, right? There are only a couple gaps here where the K-mer matching dropped below the threshold, and so it didn't put a dot there. So and this top left, this is the best pair, the most similar two sequences. So that's great. But then as you move away, you get stuff like this. And so what do you think is going on here? What is that plot, right? This one right here. Somebody want to take a stab at what does that mean? Interpret that for me.
Jeff, quick question. Can you explain what the axes are, like the GI and the...
Those are the FASTA headers, so something about the gene name. It's telling you which sequence is on the x-axis and which sequence is on the y-axis. So if you wanted to pull those two out, you could go to that p450.fasta and search for those headers, and that would give you the sequence.
Mm-hmm. Is it plotting the alignment and K-mer matches between those two genes?
It is. And so this example is ones where these two genes match really well, basically along their whole length. These two have kind of this weird pattern where you've got these two parallel lines. And so that suggests that they don't line up one-to-one along their whole length, right? But like, dig a little deeper in terms of what is it showing you? If, right, this almost solid line is saying that from this gene, here, let me click on it just to make it a little bigger, right? For the gene that's on the x-axis, from roughly 500 to 1,000, so the second half of that protein, it matches almost identically over almost the whole length of this one, which only goes to 500. So the one on the y-axis is only 500 amino acids. The one on the x-axis is about 1,000. The match is really only good for the back half of the x-axis protein, okay? And you've got basically little sporadic matches for this one. It's a similar pattern on this one. You've got a little bit better match on the front half of the x-axis protein, but there's still this weird thing where the back half is way better matched to its closest partner. Any hypotheses? Yeah, that would be my guess, right? It looks like, partly because these things are parallel lines, that the one on the x-axis actually has two copies of the one on the y-axis. It's just that the first copy of it has a terrible alignment, right? It's not very similar. And now I say it has a terrible alignment, it's still got what sort of looks like the same orientation. It's just diverged so much that the Kmers aren't the same, and so the dots it doesn't make a dot there. But follow me there? So, I talked about how P450s are these oxidative catalysts, and they recognize all kinds of molecules. And so, the intuition for me is that there's probably a conserved domain that you can't mess with in order for it to do its oxidative function. But if you duplicate that thing, part of that domain can just kind of evolve to either be bound in a membrane differently or recognize a molecule differently. I don't know. These are, just sort of hand-wavy hypotheses. But if you're really interested in P450 evolution, that's what you might do. So for our purposes, what's very clear is that there are big chunks of these proteins that don't align very well. And so if we wanted to make a phylogeny of the P450 genes, remember back to our fundamental hypothesis that every site is homologous. Well, when you've got regions of sequence that don't align basically at all, you just want to throw those out, okay? Because we don't want to assume those are homologous sites. So that gets us to the trimming. So there are lots of tools to do trimming. Trimall is, it works in colab, and it's I see it cited fairly frequently. Lots of workflows have trimmers built into them. So when we do RNA-seq later, and when we do variant calling, you'll see one called Trim-O-Matic. We've already got it installed. I've given you the link to go look at it in detail if you like, but you can also just click play here. And it's going to take our aligned FASTA file, the one that MAFFT just generated for us, and it's going to output trimmed sequence. So what it's going to do is it's just going to scan along the proteins and the multiple sequence alignment and look for blocks that are not very well conserved across the whole thing and trim those out. So that one runs pretty fast, and you should see it generated a few more files. So we've got our P450 trimmed.fasta. That's what we're going to work on from here on out. But it also generated this trimmed.html because I used the HTML out flag in there, or option. If you download that, it doesn't auto download. So I download that to my downloads, and then I'm gonna open it, maybe, there we go. So it'll open this in a browser. This is what Trimall output, and this is what it was using in order to decide which sites to throw away. So it's everything that's not in dark gray, it's gonna throw out. So it's got the selected columns, that's the gray, and then it's showing you gap scores and similarity scores. The darker the color, the better. Okay, and so you can sort of map on what it's keeping. And you can see there are these big regions. And if you remember looking at your NCBI viewer, you saw this too, right? When it was totally zoomed out, you had these little gray pieces that lined up with long stretches where there was no sequence in the majority of the proteins. And so that's what you're seeing here. But then you get down to the back half and you start to see, oh, there's actually quite a bit that aligns down here. So you're going to keep most of these regions because they align pretty well. All right, so going back to the colab notebook. Now we're going to rerun MAFFT, but we've modified it to not run on the original P450 FASTA. It's the P450 trim. So we're going to rerun our alignment on the trimmed data. Any questions? Anybody's not working? This is the beauty of pre-baked. Just hit play. But also, you know, you lose some depth of learning when it's pre-baked because you don't learn to deal with when it doesn't work. So one thing to notice, right? Remember the auto chooser? It picked a different algorithm this time. So it picked one that is more accurate but very slow. And that's because we shrank the sequences by probably half. Not in number, in length. Okay, and then the next one is just going to rerun the dot plot code, but on the new alignments. And what you'll see is that all of them now, the top 16, at least they're all on a single diagonal. We don't have any repetitive stuff. This is a perfect example where you've got everything along a diagonal, but there are these breaks that offset the diagonals a little bit. What are those? What's the biology of that? There's not a match except, so, right, think of these, there's a perfect one-to-one Kamer matching along the diagonal, and then suddenly the x-axis sort of jumps up in terms of there's no match with y, no match with y, and then it picks up again. So what happened to the sequence on y? An insertion, right? So there's an insertion there that doesn't match up with what's on in the X protein. And so then it starts matching again, and then you have another break, and it's hard to tell if that's exactly on the diagonal or not. But that's the classic insertion deletion pattern where you get a diagonal that gets shifted over on one of the sequences. And again, I talked about this last week, you can't tell when you're only doing two sequences whether that's an insertion or a deletion because there's no polarization unless you know the ancestral state. Okay, so if not good, certainly much better alignments. Okay. And so then the last section here is to create a gene tree. And so this will give us a visualization of how are all of the genes that we aligned related to each other. Okay, and there are lots of things we could do to try to make this prettier. I have sort of color coded it at a pretty color coded the clades at a low levels just to highlight families of P450 genes. Later today, when we talk about BLAST, we'll talk about homology and how to infer orthologs versus paralogs, and we'll talk about what's really going on evolutionarily and what we could infer from a tree like this. So what this is saying, right, is that there's one really big prolific family that shares a common branch, and then there are a few small families. And right, again, if you were really interested in P450 function, you might go in and say, okay, what's the consensus of this family? What's the consensus sequence of this family, where you line them all up, try to infer the ancestor of that thing, look at their function. Looking at their function is an experimental question. What do they bind to? What are their ligands? What do they catalyze? Any questions about alignment? Super common, very important early step in lots of workflows. Okay, so we're going to move on to talking about BLAST. So you'll want both of those PowerPoints, but we'll start with the overview one that talks about how does BLAST work. So I put this slide up first for multiple sequence alignment. Doing MAFFT and muscle skipped a step. Right? I gave you the P450 FASTA file, but how did I get that file? I got it actually from a publication, but if you were starting this, and the example that we're going to walk through is we just want to know if the dosage compensation genes from Drosophila are present in the beetles that my lab works on. So to do that, you go to one of these databases to look up sequences. So there are a few major repositories for DNA and protein sequence. Hopefully, most of you have some interaction already with NCBI. This is the main one in the US. I put all caps, the primary. If you publish with new DNA or protein sequence data, you're required to put it in NCBI. Okay, and there are lots of tools, and they're constantly evolving to access and to query these databases. In Europe, the main one is Ensemble. There are lots of cool tools. To really get the most out of your data, it's good to learn to use both. Something that I would say not everyone, even in the field, knows. There's an Ensemble metazoa. There's also one for protist, bacteria, fungi, and plants. So the base Ensemble, if you just search Ensemble and go to the main website, it's very vertebrate-centric, really human and mouse-centric. So if you work on something that's a non-model system, and especially ensemble metazoa, well, I should say a non-model animal, and then if you work on one of these other major categories, there's another site for you. These NCBI and Ensemble, the data, the actual sequences are mirrored every day, and then they're also mirrored with the main repository in Japan. So in principle, all the data underneath is the same. Now they've built different tools, things like we're going to talk about with identifying gene families. Ensemble has some great tools. NCBI and the Japanese database, not as much. They are increasingly integrated by links, though. And then you get more specialized databases like OMIM, which are really just focused on human and the variants that have been associated with disease. So that gets us to BLAST, Basic Local Alignment Search Tool. This is probably the most common used software in bioinformatics and maybe all biology. The question mark there is only for the all biology. The need for BLAST, so you don't get, with 129 sequences and how fast muscle and MAFFT work, you don't get a real taste for how computationally intensive multiple sequence alignment is. So if I have a sequence of 500 or 1,000 amino acids, and I want to find all of the things that are similar to it that have ever been sequenced, you can imagine like millions and millions and millions of sequences that you have to search and do alignment for, okay? That's not computationally feasible. And so that's what BLAST solved. And it's called a heuristic search because it's not guaranteed to find you the best alignment. It's guaranteed to find you some similar sequences, okay? So it's an important distinction. It will give you alignments, but they are not guaranteed to even be the optimal alignment with the sequence that it retrieves, okay? So how does that heuristic method work? So this protein sequence up at the top is what we're going to pretend we want to find a match for. The first step in BLAST is that it splits the query, and so that sequence that we are giving it that we want to search with, that's called the query sequence. It's going to split that into, in this example, the word size is 3. And so, when we talk about word size for BLAST, we're talking about how long are we chopping up our query for to go look in the database. If you want to find things that are more similar, you make your word size bigger, okay? Because then it's, as you'll see, that is the foundation of the search, is each individual word, okay? So our query length is 11 amino acids, and our word size here is 3. Next step is to generate the neighborhood of words that have a score less than some arbitrary, not arbitrary, it's a threshold that you can set. And so if you worked through all the Rosalind problems when we were doing the homework from 2 weeks ago, right, with the picture and the comments, one of those steps was to generate the D neighborhood of a string, okay? So everything that is a certain Hamming distance away from all the k-mers, that's what this is doing, exactly. So except for amino acid, we only dealt with it for nucleotide sequence, where every step only had 3 other possibilities. Okay, so this, for the amino acids, you've got 20 possible things at every position in the word. So you generate that neighborhood, and then you use Blossom 62. This is the default. You can change that. It's a setting. And you go looking through the database. This is the thresholding thing. Which words are we actually going to go look for? Only words that are above a certain threshold. So things that are like, especially in amino acid space, not all of those 20 to the third potential neighbors are likely enough to bother with. Okay, so we're going to say anything that's below a certain threshold, we're not even going to use that as a search term. Okay, so that's the first sort of speeded up step, is that we're only going to use a certain number of words, certain number of neighbors to our words that are above a certain threshold. Okay, so that's the word list, and then it takes that word list and goes and searches all the sequences that have ever been sequenced, at least by default. Okay, we'll talk about how you modify the search space in a second. Okay, and what it's going to do is, for each of those words, it's going to find matches in the database that are embedded in other proteins, and then it's going to pull out those sequences and start to extend. So QEH was the match to it's one of the words in our search list. The matches get a score of 5, and these are Blossom scores. And so it's an H match is an 8, okay, K's are 5. Then we've got our first mismatch, -1, and a G match, and then that mismatch is a -3. And so the way to think about this is the QEH, our score of our alignment, is increasing, increasing, increasing. It drops, but then it doesn't drop enough. So the maximum drop-off score is another tunable characteristic. So dropping off by 1, like this mismatch isn't enough to stop extending this match. Okay. It goes back and matches another one, but then an S to W is a big enough mismatch that you're going to stop extending right there. Okay. Then, so it takes that section all the way out to the GG match, and it starts extending in the other direction. And in principle, it does this at the same time computationally, but here, right, we go from Q to C, still going up. It drops a little bit on this mismatch, but not below our cutoff. Matches again, and then it has a bad mismatch that drops it off. So we trim going the other direction at that YD mismatch. And the result of that algorithm is that you retrieve maximal segment pairs. And so when it gives you alignments back, it's really giving you these alignments between maximal segment pairs. The score for that, we'll talk about the bit scores later. This is not, this score does not depend on the database or the query size. This is just how well does your query segment match with the thing that it found in the database. So then it sorts all of the maximal segment pairs. And keeps the ones that are above a threshold. You can tell it how many to keep, like only keep the very best one, which is what we'll do on Thursday when we want to look at for orthologs. And then a couple of statistics that are important to keep in mind when you're talking about blast. Probably the one that gets cited and looked at the most is the E value. That's the number of maximal segment pairs that are expected to have similar or higher score by chance. This is the function for how the E value is derived. It's not like I'm going to ask you to derive E values, but I think it's worth understanding like what determines the likelihood of finding a maximal segment pair match just by chance. Like you're just sprinkling nucleotides or amino acids down by chance. What's the random chance expectation? Because we are searching millions and millions of sequences. And that's determined by how long is the query and how big is the search space. That determines the probability of finding a match like that by random chance. Okay, so conceptually not too difficult to wrap your head around, I think. What that means, though, is that you think about if you've got a really small query and a really big database, there's a pretty good chance you might find something just by random chance. Okay, like think about it in the extreme. If I have one amino acid and I go search the database, I'm going to find it all over the place. You get a little less likely if you add another amino acid. If you keep stretching that out, it becomes very unlikely. Okay. Same thing where if I've got a amino acid that's 10 amino acids long, protein that's 10 amino acids long, and my database consists of one sequence, pretty unlikely I'm going to find that by random chance in that database. But if the database has millions of sequences, still some chance. That's what the E-value is measuring. Okay? And so this box is saying, whoops, it's cut off on the, right. If you can restrict the database, because usually you can't restrict your query sequence. That's the thing that you want to go find. Find matches to. But you can often restrict the database. So in our example, we're going to restrict it from looking at all sequences to only the sequences that are in Tribolium castaneum, so the beetle, for example. So that will shrink the database, making things less likely to be seen just by random chance. The bit score, so I was talking about how this is the score for this alignment. Well, it depends on the length of the segment pair, which if you want to compare across all alignments, and especially across different queries, you need to normalize that. So that's what you really just need to know, is that the bit score is a normalized version that you can compare across different searches. So what I mean here, the E-values you can't directly compare. Like an E-value of 0.001 for one search that I did is not directly comparable to 0.001 in another search that I did, because the queries are different and potentially the database is different. So that 0.001 means something different to those two queries. But the bit score is comparable because you can't change how well the thing aligned to the other sequence. Okay, so the alignment score is something that you can rank across searches. The E-value, it still tells you whether or not it's likely or not to be random chance in the given search, but you can't necessarily extend that to comparing to a different search. Okay, but you have it in notes, and that's important stuff to understand when you're thinking about what do my blast results really mean. All right, so let's do some blasting. The next. Oh, I forgot to take off the homework part. I'll fix that later. There's no homework for this one, for blast and NCBI. We're going to do blast on bridges on Thursday, and there will be homework for that one. So the example that we're going to play with is, this is the dosage compensation machinery. One of the questions, like really basic questions that my lab is interested in, is how do males get away with having only one X chromosome? If you do that for any autosome, lethal, okay? But males get away with being haploid for the sex chromosome all the time, okay? And so in Drosophila, the way they get away with it is that this dosage compensation complex comes in and helps hypertranscribe all the genes on the X chromosome. So even though there's only one copy of all those genes, they get expressed at roughly double the rate of an autosomal gene. Okay? So that's what we mean by dosage compensation. It's compensating for being haploid. This machinery is not conserved in all XY species. Mammals don't have this. Okay? So we get away with it in a completely different way. Okay? But Tribolium, in terms of the pattern of X expression, looks sort of similar to Drosophila. The question is, are they doing it mechanistically the same way? Are all those same proteins conserved from fly to beetle? Okay, so biological question, how do we address that computationally? Well, let's go look and see if there are homologs to all of those proteins in the Tribolium genome. So first step is, if you go to NCBI, for the sake of time, just so you have time to play with this, I'm going to just keep walking through the pre-baked screenshots here of what you would see. So we would go to NCBI. You can follow that link. We would pick the gene database and give it the name of our gene of interest, which the first one on that list is malus. You could also give it the gene ID, which is a unique number. And I also highlighted the fact that I'm logged in. So if you created an account on NCBI, you can save your searches and go back later and retrieve your search results. And the cartoons here are, there is so much data in NCBI. We want to, at any step we can, limit the search space. Okay, so that's why we're picking just the gene database. If we just searched all of NCBI for malus, we would get a lot of hits in different databases to have to search through. The result page, if you scroll down a little bit on the initial result page for malus, you'll find this graphic. And MLE is the abbreviation for malus. It says there are 4 accessions, which are just the entries. Accession numbers are entries in NCBI. If you click on that, where the arrow is pointing, it pops open. And I should also add, a lot of times if you aren't sure where something is in NCBI, if you hover over it with your mouse, it pops up a menu. So clicking on that will open up the 2 purple and the 2 red. Those are the RNA and the translation, so the protein version, for each of the 4 things that were under MLE. Why are there multiple? Because there are different isoforms of malus. And so when we search, to see if there's a homolog in Tribolium, a good strategy when you have multiple isoforms is to pick the longest one. So if you the P450s are a good example where you've got some parts of the protein that are really well conserved, and if there are different isoforms and you pick a short isoform, there's a chance you'll pick a part of the protein that evolves really quickly. So just as a default strategy, always pick the longest isoform if you're going to search, especially in some other species. So hovering over any one of those this is what I was talking about, where you hover over it and it'll pop up a menu. And it tells you all kinds of statistics about whatever accession number you're hovering over. You could link straight to a protein blast, which is what we're going to do, straight from that graphic or from that pop-up menu. This pop-up also tells us the gene ID. So if you go back to this, and you actually, when we start to play with the other genes, make sure you're looking at the right gene ID. Gene names are one of the banes of computational biology's existence. They change, they're not consistent from species to species. Some genes have multiple names because they were identified by different labs and named by different labs. It's messy. So gene IDs are not, though. So in principle, to the extent that things are well curated, and if you're working on a model system, there's a better chance. Even if male-less is an easy example because it doesn't really have multiple names, at least in Drosophila. But the number will correspond to all of the synonyms for that thing. So if there are multiple names, at least if you're looking at the same number, you should be talking about the same gene. There are lots of ways to get to BLAST. That pop-up menu, clicking it will take you straight there. You can Google it. Almost every page in NCBI has some way to link to BLAST. But getting there will take you to a page that looks like this. We're going to do protein BLAST. The accession number for the longest male-less isoform was this, NP476641.1. If you clicked this button, right, so if you click right there, it automatically takes you straight to this page with that number filled in already. Whoops. Sorry. The database that we're going to search is the non-redundant, and this, I think this has changed. I didn't update the screenshot. It's still called NR, but I think the verbiage on what they're calling that now doesn't matter. NR is the important part. It's the non-redundant database, so they've taken out all the redundant sequences. And again, this is that fire hose example. We're trying to limit the search space a little bit. And then you could hit BLAST right there. We're assuming, right, I can't scroll down on that because I'm on screenshots, but making sure that BLAST P is selected and then BLAST. What you would get in that case is hits to Drosophila melanogaster. So there are lots and lots and lots of sequence for male-less gene in melanogaster from different strains of Drosophila melanogaster, different variants, so mutations in MLE get their own accession number. If you've done an experiment and you've sequenced MLE and you published it, You have to put those sequences in NCBI. Okay. Okay. So we're not really interested in is Malus in Drosophila though, right? Because we already know. We pulled it from Drosophila. You know, somewhat reassuringly, we got a 100% match over 100% of the query, so that's all good. And this is just a slide to tell you what the columns in your output table are telling you. But now to the, right, what we actually want to do is go look in Tribolium castaneum. Now, if you scroll down far enough, I've cropped the screenshot here. If you were to just keep scrolling and scrolling, this is everything that matched Malus. It just happens that, you know, the whole, you know, first 5 pages are all Drosophila, okay? But eventually, if you scroll down far enough, you would find Tribolium. But alternatively, right, make this database search space smaller. It will improve our e-values, right? What, you know, our probability of getting a match by chance. And if you do that, you should get the page that looks like this. Okay. So there are a couple different isoforms of the same gene. Okay. And the best hit here, right, saying the maximum, this is the bit score, is, again, you can compare these to each other. You can compare them across. The query coverage, 91% of the Drosophila Malus gene, that's the query, is matched by the Tribolium gene. But those matches are only about 60% identical. Okay. So what does that mean? Thinking about the alignment, that means that we've got amino acids that are lined up about not, and you can think about it in our dot plot too, about 91% of the length is right on the diagonal. But the dots, you know, only 61% of the sites actually get a dot for being identical. Okay, so it does look like some of those P450s where you had dots on the diagonal, but there were a lot of space in between them. Okay, and this is real biology. Then the question is that gene in Tribolium the same gene that's in Drosophila? Is it functionally similar enough that it's doing the same thing? So you can download the description table. You can download the FASTA versions of all those sequences. If you click on the graphical summary, so that's one of the tabs across the top, you can see that those top two, and the red means highly similar. You get pictures of the domain structure. A lot of cool information just a click away there to look at how well do those things align. And then that question about are they the same gene, can we infer that they're homologous? It's a messy question, and so I've given you some reading. This introduction to sequence similarity talks about rules of thumb, because functional homology ultimately is a wet lab question, not a computational question. The best I can do is, you know, even if I tell you it's 100% identical over 100% of the sequence, yes, that is the same gene in this other species. I still can't tell you if it's functionally doing the same thing, okay? Because I don't know expression timing, I don't know expression location, I don't know if it interacts with some other proteins in important ways. So that sort of gets to the limit of what this computational homology search on sequence can tell you. All right, so for the last 10 or 15 minutes, turn this from homework into practice. That should say is. Look at the other 3 genes in the dosage compensation complex. That's male-specific lethal 2, lethal 3, and males absent on the first. And I've given you gene IDs there. And just do that blast to see what the tables tell you. And just because we live in the age of AI now, you can ask ChatGPT to do this for you and see what its limits are. Oh, come on. And I'm going to tell it to use the longest isoform, use protein blast. Anything else I should tell it? I already told it to look in tribolium. See what it tells me. I already know ChatGPT can't do blast. But it can do some really cool stuff. It'll tell you what the genes do. If it's been annotated in the other species, it will tell you, and right, this has given me a candidate already. And so it basically said, yes, it has one, and then it gave me the gene that I can go look up. I can also tell it to give me yeah. Let's try it. Whoops, I misspelled Drosophila, but it's smart. I was going to phrase the question differently, but what it usually tells me is I can't blast, but here's the link to run the blast. Mm-hmm. Oh, did it? I guess I didn't. I wasn't reading. Oh, yeah. So it says it can, but then if you tell it to. Listen, yeah. Maybe it did. Even if it does, we're still in a situation where I would always go run it myself. That's the nice thing about having it give you the URL for the blast. So I can go back while it's working on that. I can show you conversation from earlier. This is what I mean by like when I told it to blast, what it did was say I can't actually run blast from here, but I set you up with a one-click blast P, and so you can just copy and paste this into a browser and it will run the blast for you. So it does save a few steps of having to go find the gene, get the sequence, and do the blast. And right, this is the same one that I was showing in the screenshots. Right? It recognized the right isoform to use.
The MSO2 has two isoforms there, isoform A and B.
The longest one. It's interesting. It looks like it is doing the blast. Which surprised me it surprised me that it couldn't earlier. So I asked it to give me the blast output so I could look at the table, and it said I can't run the live blast, and then it gave me links. So I'm opening the link to clustered NR. So clustered NR is the new subset of the NR database. That's the default. It's interesting. It's not pre-filling. Love hate with AI. So what is anybody finish the MSL2 example yet?
Put the gene out there into just like the NCBI pulls it up, but it doesn't give you any of the proteins. So you can't run it against or running blast.
Go gene. So did you get to this step? So doing the same steps, I went to gene and I pasted in the number in this case instead of the name, which male specific lethal 2 or MSL-2 should also work. Then I scrolled down to where this was, popped open the different isoforms. These proteins, and again, if you have a choice between searching nucleotide or protein, always pick the protein because more information. That's 773. 773. So, right, I'm looking at the protein length here. So these two isoforms are the same. It's really just the 5' UTR that's different. So there's some regulatory information maybe, localization information, who knows. So it shouldn't matter which one of these red lines, which is the protein that I mouse over. And then I can click down here on blast protein for that accession. Pick tribolium, blast away. So I'm going to go back here. Some of the stuff that I talked about in the how does blast work part of lecture, you have to expand the algorithm parameters, right? So things like word size, the expect threshold, all of those things that I was talking about that are tunable, you can find here. You can use a different scoring matrix, different gap cost. Remember when we talked about pairwise alignment in terms of inserting a gap, like starting a gap versus extending a gap? They've done a really cool job of putting these question marks in, so you can, I don't know if it's a mouse over or a click, but they pop up little explanations to, jog your memory for what that parameter, why you might want to tune it, and tune it to what? Does that solve the problem? There are lots of ways to get the same thing done in NCBI. Some of the, like, I am showing you or at least one of the efficient ways, I think, but searching and Googling and, you can, what I mean is you could download the FASTA sequence for male-less 2 or male-specific lethal 2, and then paste that FASTA sequence into here, or upload a file with that FASTA sequence, and then run the blast. And you would get to the same answer, okay? It's just a little bit less efficient than the way that I was showing you, okay? But it's the same thing. And I often see people copying and pasting long sequences into this window rather than the accession number. Don't do that. In principle, it gets you to the same answer. It just opens the opportunity for mistakes because you end up not copying the whole sequence, right? So you accidentally truncate it, and it's long enough that it went off the window, and so you didn't know that. These are like pro tips. But many pro tips you have to experience for yourself before you actually learn them. All right, we're out of time. So again, you can play around with that. We'll start out Thursday. I'll ask you if MSL or the dosage compensation complex is conserved or not. So it'd be fun if you've looked at them all and know where the problems are, like in terms of interpretation. And then we will do blast on bridges with big data sets. So, okay.